Okay, so I'm going to go ahead and get started. Good afternoon and welcome to the Psychology Department Speaker Series. My name is Julia Boehm and I'm an Associate Professor of Psychology here at Chapman. I have the pleasure of introducing Dr. Laura Glynn, who is today's invited speaker. Dr. Glynn received her PhD in experimental psychology from UC San Diego. She then completed a postdoctoral fellowship at UC Irvine. Dr. Glynn joined Chapman's Psychology Department in 2008 and has been a leader here ever since. So Dr. Glynn's research focuses on how psychosocial, behavioral, and biological processes during pregnancy impact outcomes for both mothers and children. So her work involves assessing women during the prenatal period and then following mothers and their children across many years. So working with these longitudinal cohorts, she has made discoveries about how cognitive function changes for women during and after pregnancy, as well as how factors that are associated with premature birth, among others. So her program of research is funded by the National Institutes of Health. With more than 10,000 citations of her work, Dr. Glynn is one of the most highly cited professors at Chapman University. I'm delighted to have Dr. Glynn here today to share her insights into how unpredictable patterns in mothers' mood and behavior may be related to the mental health of their children. Now, to give you a sense of how we will proceed, Dr. Glynn will give her presentation for the next 40 or 45 minutes. If you have questions during her presentation, please feel free to include them in the chat. After her presentation, Dr. Glynn will address questions from the audience. So please join me in giving Dr. Glynn a warm Zoom welcome.
Great. Thanks, Dr. Bain, and for inviting me and for that very nice introduction. I'm really excited to be here virtually with you all to share some of my work today. I do want to note, if you see me looking over here, I have two monitors and so I can see my slides here, you all over on the right. So if I'm looking, that means I'm actually looking at the audience. Okay, so we'll dive right in. And for the past 20 years or so, a primary component of my research program has focused on exploring the developmental origins of health and disease. And the central premise of this conceptual model, the DOHAD model, is that early life experiences, including early adversity, beginning in fetal life, have profound implications for trajectories of health and disease across the lifespan. And while the field has focused broadly on tying these early experiences to a range of physical health outcomes, I've been focused, among those, focused primarily on mental health trajectories. So that's what I'm going to be talking to you about today. In terms of these early life exposures, the DOHAD field in general has focused on how things such as traumatic exposures, poverty, abuse, and famine shape developmental trajectories. However, and it's hard for me to believe this, for almost a decade, I've been collaborating with an interdisciplinary group of researchers who are part of an NIMH-funded Conti Center focused on a particular type of early life adversity, which is unpredictability in the early environment. And so today we're going to talk about why we might study unpredictability as a novel form of early life adversity. I'm going to be providing you evidence that is a potent influence on cognitive and emotional outcomes. In this conversation, we're going to take a ecological approach and examine unpredictability across multiple contexts that are relevant for the developing child. I'm going to begin by talking a little bit about unpredictable inputs at the parental level. We'll then move out to the home environment and finally end up with a quite broad measure of unpredictability in the early environment and look at the influences of that. I'll also be speaking just a bit to potential biological mechanisms or biomarkers, a little bit in the domain of neurodevelopment, which is a focus of this group. I do have some really exciting findings in the domain of methylomics or epigenetics, but in the interest of time, I'm not going to speak about that, but I did just want to note that is something that we're pursuing in this work. In terms of how we're attacking these problems, we're really using two complementary approaches. And the first is a cross-species approach. So we're looking at these questions in both non-human animal models and in humans. The great advantage of this is that obviously all of our human work is correlational, whereas we can experimentally manipulate exposures to unpredictability in the rodents, which really do allow us to draw more confident conclusions about causality. And the other big advantage of this cross-species approach is that the rodents obviously allow a deeper and more detailed probing of neurobiological mechanisms of interest. So in addition to leveraging these cross-species approaches in my lab at Chapman, I've been following, as Julia mentioned, 2 prospective longitudinal cohorts of Southern California children with over 200 kids in each. In each cohort, we recruited women in the first trimester of pregnancy. Then in both cohorts, mothers and their children are followed intensively through infancy into childhood, and in the case of the older cohort, into adolescence. And currently in my lab, we're assessing the older cohort at 16 to 17 years of age, and have just begun seeing the kids at 18 to 20 years. In fact, you can see that's so new that I don't even actually have a lab picture yet, I realized, of the kids at those visits. And in the younger cohort, we're seeing the kids at 3 and 5 currently, and we will be doing our first 7-year-old visits at the end of this summer. So that's where we are in terms of these prospective cohorts. And I did want to note before diving into unpredictability that not only are these children and their mothers characterized carefully over time, but we also have intensively characterized their developmental trajectories with a wide range of developmentally appropriate assessments. The assessments of child development begin when the children are fetuses with in utero assessments of neurodevelopment. We do a very broad range of behavioral assessments like mother-child interaction, coding of infant temperament. And as the kids get older, we can do more things with them, including clinical diagnostic interviews, more intensive cognitive assessments, and biological sample collection, and neuroimaging. So for example, we start that at about 6 to 7 years of age. So that younger cohort I told you at their 7-year visit will be the first time that they'll head into the MRI. So that's a broad view of what's going on with the human participants about whom I'm going to be presenting data today. I also wanted to overview for you the preclinical or non-human animal paradigm we were relying on in this work. And this is developed by a collaborator at UCI, Tally Barham. And what she has done is develop a paradigm that simulates poverty and chronic early life stress with something called the limited bedding and nesting paradigm. And you can see here on the left, a control mother with her pups in her cage. She's got plenty of sort of soft surface to walk around on and plenty of bedding and nesting material for her pups. In the cage on the right, you see the mom who's in the LBN paradigm, and you can see that she has kind of an impoverished cage environment where she's given very little to make her nest with and doesn't have that nice sort of soft surface to start raise her pups in. And this is in this paradigm, the LBN or limited bedding nesting condition lasts from days P2 to P10, so postnatal days 2 to 10 for anybody who's a rodent person. And we know now through Tally's work primarily that when this experimental manipulation is imposed during this sensitive period, it has long-term consequences. So we now know, for example, that is associated with cognitive problems in offspring raised in this condition, including deficits in memory and epigenetic reprogramming of hippocampal neurons. We also see problems in the emotional domain, including reduced ability to experience pleasure and also sensitivity to stress. And what's interesting about this paradigm and why I'm talking about it and what makes it relevant to unpredictability is what we know about it, what this paradigm does to maternal behavior. And what it does specifically is induce fragmented and unpredictable maternal care. So I'm going to show you a video that illustrates this, and I want to just orient you. On the top, you're going to see the mother who's in the limited bedding nesting condition. On the bottom, you're going to see a mom in the control condition. This is obviously time-lapsed, and I think even if you're someone who doesn't study rodents, you can probably pretty easily pick out the differences in the behavior going on in these two. The mother who has sufficient nesting and resources is able to provide nice long bouts of nursing and licking and grooming, whereas the mom on the top, her movement is quite scattered and divided, perhaps a bit the way many parents may feel during the pandemic. I know I've certainly felt this way myself. And so again, you see that sort of fragmented behavior going on in the mom and the LBN condition. So clearly the patterns are different. Here are those data quantified. In rats, when we talk about maternal behavior, we look at things like licking and grooming, nursing, time spent on the nest. And regardless of which of those behaviors you look at, what you see is that the duration of those bouts of maternal behavior are much shorter in the LBN moms. What's really interesting is that the actual amount of those behaviors does not differ between these two groups. So while it is the case that the LBN moms are doing more fragmented or shorter, unpredictable bouts of licking and grooming and time spent on the nest and nursing, the absolute levels of inputs for these two groups of pups do not differ. So this sort of harkens back to this idea of the importance of patterning of these behaviors as opposed to the absolute levels of behaviors themselves. And that's something we're going to be talking about quite a bit today. So the question then becomes, what about humans? We have, again, through the work of Dr. Barham, this pretty solid literature linking this fragmented care to development in these rodent models. But what about in humans? Is it the case that patterns of these sensory signals may impact child development similarly? And a reason that we think this is due to a couple of things. First of all, there are these classic experiments from Hubel and Wiesel and the more recent really elegant work of Takao Hench. And we know that patterns of sensory signals shape the development of sensory systems. So for example, exposures to patterns of light and sound, tones are required for proper maturation of visual and auditory systems. And our group is positing that the same principle applies to cognitive circuits, and that the maturation of these circuits might require predictable and consistent sequences of environmental signals from the mother as well. And as I said, we do have evidence from rodents that unpredictability of maternal signals impacts stress responses and memory. So to address this question in humans, what we did was in both cohorts, we have assessments of mother-child interaction in the lab when the infants were 6 and 12 months of age. In this paradigm, mothers are given sets of toys and told to play with their infants as they would at home, and we record these interactions. In these interactions, we then code maternal sensory signals through real-time continuous observation of these behaviors during a 10-minute semi-structured play episode. And I'm going to walk you through what we're doing in the humans. Essentially, we're coding sensory signals in what we think of as 3 channels of input. One is auditory. So anytime mom makes an utterance in that time period, there's a tactile channel where we're looking at different types of touch, holding, carrying, restraining, and supporting the baby. And in the visual channel, we coded instances where mom is manipulating an object and both she and the baby are visually attending to this object. So we have 3 channels. We go through and code this continuously throughout that 10-minute interaction. Here, what you can see is a visualization of those sensory signals. So on the top are the instances of auditory inputs. In the middle, you can see tactile, and then on the bottom, visual over time. And you can notice that the auditory, for example, is much more common than, say, the tactile inputs or occurrences. What we then do is look at these in conjunction, because we're interested in transitions between these states. So what you see here in this figure is a continuous time series analysis of maternal sensory signals in this context, using 8 possible categories of inputs, recognizing that mom can engage in a combination of sensory signals too. So for example, at the top are the instances for this mother in which she was doing auditory, tactile, and visual concurrently, all at the same time. In the next row, you can see instances where visual and tactile signals were happening at the same time. The next third row is auditory and visual together, and so on. And so it's also the case, of course, that at the bottom, you can see we recognize that she may be giving no input to the infant at all at a given moment. What we do next is convert this into a discrete time series of maternal sensory signals where the signal has been reduced to a single dot. So we're not considering duration here. This is just an instance. So we can look at transitions. And this is, as I said, what we're interested in, the predictability of these signals or transitions. bear with me, we're almost there to this summary score we're going to use to predict outcomes. And I just want to take a moment. I am going to be throughout this talk calling out trainees associated with this Conti Center who really drove parts of this work forward. And the first of those is Brian Vegetable, who is no longer a trainee, but when he was a PhD student working with Hal Stern at UCI, he's focused a lot of his dissertation on helping us to develop these indices of maternal sensory signals in human and rodent moms. So we have those transitions I just showed you that we can, from that, a matrix can be used to organize the transition frequencies with rows indicating the initial behavior for each pair of actions, and the columns, the behaviors that mom is transitioning into. So for example, 65 is the count of times that that mother transitioned from a visual to an auditory visual cue, for example. What you see in this matrix is the probability of each transition type, which is the proportion of time an event follows another event. And we can consider this the empirical transition probability from the row event to the column event of a first order Markov chain. And again, this is Brian's really great work. And I also wanted to note that for anybody who's interested in these measures that I'm talking about today, both the coding scheme and the R code to develop this is publicly available on the center website. So in a parallel methodology, we did the exact same thing to characterize predictability of sensory inputs within the rodents. Here we observed dams for 50-minute windows twice a day for 8 days, looking at licking and grooming, carrying the pups, nursing, and all the things that rodent mothers do. And again, we can then index predictability of maternal transitions across sensory domains. So last, we do want a summary variable that we can use to predict outcomes, which is an entropy rate, which is a measure of how random or unpredictable a mother's next behavior would appear to an observer who is making a guess based only on the most recently observed behavior. And what I really want you to take away from this, all of this, is that unpredictable sensory signals are associated with higher entropy rates. And in humans, you can see on the left, we see this as a distribution of entropy rates because we're just looking at naturally occurring transitions and behaviors in these moms. And of course, in the rodents, we have groups because we manipulate this with our experimental paradigm. And you can see again that the rodents who are in the LBN condition in purple on the right have higher entropy rates when we apply this coding scheme to the moms. After all of that work at the front end, we've now linked unpredictable sensory signals to less optimal child development beginning in infancy. Here you can see an example of finding from a cross-species paper in which we show that in both 6-year-old children and in rats, higher levels of unpredictable sensory signals are predicting poor recall memory performance. So I think you may remember that I said we knew prior to initiating this work in humans that memory performance is one of the things that was reliably affected by this LBN manipulation. And here we're seeing again that entropy rate or the unpredictable maternal signals are predicting poor memory performance across species. In terms of the humans, after working on this for a while, I think probably the strongest and most consistent findings we have are for those related to self-regulation or effortful control. And specifically, we've shown now repeatedly that children who are exposed to more unpredictable sensory signals show lower levels of effortful control. And this is important because there's a really extensive literature establishing poor self-control as a potent risk factor for psychopathology. So to go back to what we're trying to predict as an outcome here, that's what we care about. We've detected these effects as early as 1 year of age, and it's important to note that we've also replicated these findings in 2 independent samples in 2 continents. So here in my Southern California samples that I follow in my lab, but also we have an ongoing collaboration with investigators on the FinBrain project, which is a large national prospective longitudinal study in Finland, and they have also observed similar things in their cohort. Most recently, and this isn't published yet, we've linked this measure of unpredictable sensory signals to self-regulation in our adolescents. So we'll be sort of moving that forward to get out in literature soon. So I think or at least I hope that I'm starting to convince you that there may be that unpredictable maternal signals may be important predictors of cognitive development, including memory and cognitive control. But I also promise we're going to talk a little bit about etiological mechanisms underlying these associations potentially. We know, for example, that the brain is organized into circuits whose function underlies complex behaviors. And through the Barham lab with her experimental animal models, she has shown that unpredictable maternal care patterns directly induce aberrant synaptic connectivity and disturb maturation of cognitive and emotional brain circuits. And to illustrate that, you can see here data from hippocampal neurons among animals who were exposed to unpredictable sensory signals early in life versus the control animals. And I hope you can see pretty clearly that exposure to unpredictable sensory signals associated with impoverished dendritic trees in the hippocampus. And you can see that reflected both in the dendritic length and in the branching by comparing those two pictures there. And we're beginning to look at this in humans. And with the kids in my older cohort, we tested the hypothesis that more exposure to more unpredictable maternal signals in infancy. So again, taking that entropy score computed at 6 and 12 months of age, and whether that's associated with aberrant maturation of cortical limbic pathways. And this project was led recently by a PhD student also at UCI, Steve Granger. He focused on the unsnit fasciculus, which is the primary fiber bundle connecting the amygdala to the orbital frontal cortex, and it's a key component of the medial temporal lobe prefrontal cortex circuit. And you can see the unsnit highlighted there in green. And why focus on the unsnit? Well, one reason is that across species, aberrant maturation of amygdala prefrontal circuits is well recognized as a key outcome of exposure to stress in early life. And more specifically, Dylan G and others have hypothesized that accelerated maturation of the amygdala prefrontal cortex functional connectivity may be an adaptive response to early life adversity. So hypothesizing an accelerated connection here. And last, previous studies have linked development of unsnit to early life adversity, including a history of institutionalization during childhood and also to parental depressive symptoms. So using diffusion imaging, Steve quantified the integrity of the unsnit using generalized fractional anisotropy, or GFA. And what he found is consistent with those predictions of G and others, that higher maternal unpredictability during infancy was associated with greater unsnit fasciculus GFA. And so these are among our first findings suggesting that altered maturation of cortical limbic circuits may constitute a mechanism through which unpredictable sensory signals in early life may impact cognition in later life. And we're moving on to continue this work with both structural and functional as well as resting state data. Okay, so that's maternal sensory entropy. I want to now pivot to talk a little bit about variability in maternal mood. And so in addition to these unpredictable sensory signals, we've also been examining variability in maternal mood. And in addition to the background I just provided regarding the importance of patterning of maternal signals broadly, there are some additional or theoretical and empirical reasons to expect that mood variability might represent an important maternal signal for the developing child. Some of it has to do with work in personality psychology that looks at the importance of intra-individual variability and patterns of behaviors, attitudes, and feelings. For the past 50 years or so, emotion theorists have recognized that variability of mood patterns independent of their level are central components of affective experience in their own right. And we also know that increased mood variability across hours or days has been linked to risk for bipolar disorders, clinical and subclinical depression, and also anxiety disorders. Taking all of this together, these theoretical perspectives and empirical findings highlighting the link between individual differences in mood variability and risk for psychopathology are really consistent with a broader growing literature that identifies deficit in emotion regulation as a transdiagnostic factor in mental health and mood disorders. And what is really interesting here, I think, is that despite this literature that recognizes the importance of mood variability or consistency, coupled with the fact that we know that maternal mood states and psychological distress profoundly influence fetal and child development, no one is really asking the question of how maternal mood patterns might influence child outcomes. And that's what I'm going to talk a little bit about here. And we get to talk a little bit more about entropy. So with this group, again, working really closely with Hal Stern at UCI, who leads our computational core, in order to characterize the predictability or fragmentation of maternal mood, we calculated an entropy score by applying Shannon's entropy to the distribution of responses on questionnaires assessing mood. So I'll walk you through that. You can imagine, if you look at the table at the bottom, that those are the responses of 3 women on a standard mood scale. Let's call it a state anxiety scale. And you can see that a participant, like the one in the middle, who reports never worried or always secure on this anxiety scale, would be, she answers all A's, would be considered very predictable and thus have a very low entropy score. Whereas a woman who answers almost completely randomly, such as the one on the top row, would have a very high entropy score. And the third case at the bottom, where the participant is answering mostly D's and C's, would be somewhere in between the two. This is a measure that we have developed of mood entropy. It can be applied across, for those of you who are interested in study emotions and their role in health, it can be applied essentially across, as far as we can tell, any standardized assessment of mood. I'm going to be showing you data today where we computed this index from a standardized depression scale, the CESD, Spielberger's state anxiety scale, and then Cohen's perceived stress scale. And then what we did is normalize those scores and average them. And that's going to be our predictor that you're going to see coming up. And I also want to note, for anybody who wants to do it, again, the code to compute the scores is freely available on the center website. So in terms of associations with outcomes, we've been able to link maternal mood entropy to trajectories of cognitive and emotional development. Here I want to highlight another trainee, Marian Howland, who was a postbac in my lab, who's now in a PhD program at University of Minnesota, and she was really instrumental in pushing this initial work forward with me. And together we have shown that higher exposures to both pre and postnatal mood entropy are associated with increased infant negative affectivity, a finding we've replicated now in both of the cohorts that I showed you in the lab. It predicts child negative affectivity or negative temperament. We've linked it to child cognitive development, including language and more generalized measures of cognitive development in toddlerhood. And even more recently to adolescent self-reports of anxiety and depression. The most recent development here is in this really productive collaboration with the Finns. They have replicated the temperament findings in a sample of more than 1,500 Finnish mothers and children. So that's a nice development on that front. I did want to say that, or share that getting this concept into the literature was a real challenge. This was a heavy lift. It took quite a bit of work to convince reviewers, and we had many astute reviewers who really put us through our paces. And given all of this work, I would guess that I have a pretty good idea of what questions might currently be popping into your heads as you think about these findings. The first is, what do we think this is exactly? I'm calling it a measure of unpredictable maternal mood. This is something that is of ongoing interest and something we continue to work on. However, I can tell you a few things about it. The first thing is that this higher mood entropy score that I described is positively associated with mood variability collected through ecological momentary assessment. So for those of you who aren't familiar with this technique, what you do is these days load an app onto someone's smartphone or ping them on their phone, say 8 times a day over the course of several days, asking them how they feel at that moment. And then from that, you can compute measures of affective stability or variability. And it is the case that, as we would predict, women who score higher on this mood entropy scale also report more variability in their moods across days in the real world. We've also linked it to self-reports of affective lability. Again, that's whether women say they have a lot of fluctuations in their mood, and it is associated with reduced self-reports of reduced emotional awareness and a lack of emotional clarity. So I guess what I can say about this is that it is broadly, I think, an indicator of emotion or mood dysregulation, a component of which I do believe is associated with patterning of mood over time, which is what we really care about in this unpredictability context. I also wanted to talk a little bit about alternate explanations because, of course, as I mentioned in the beginning, everything we're doing in humans is necessarily correlational. And I can tell you that we've really carefully considered these more obvious and less obvious third variable explanations. And the associations between mood entropy and emotional vulnerabilities do survive analysis and consideration of potential confounders and maternal characteristics, including maternal pre- and postnatal mood levels, which is really important, maternal education, age, and socioeconomic status. And then in the case of the cognitive outcomes, we also consider mom's intelligence level. The last alternate explanation I want to address, because it is a common one that people pose, relates to, isn't this just something about mom's reporting style or her literacy level or her attention to completing these questionnaires that we're giving her? And what I can tell you to address that, what we did is we computed a similar entropy score from non-emotion measures and then look at to see whether that's predicting outcomes. And just to really convince you of that, I want to show you some data. So what you see here is that mood entropy score in blue. On the y-axis is the correlation coefficient or the strength of the association. So you can see what I already told you, which is higher levels of mood entropy are predicting higher levels of adolescent self-reports of anxiety and depression. And then what you see in green is the same entropy score calculated from a Likert type questionnaire that has similar psychometric properties to the emotion questionnaires that assesses physical activity. And what you can see is that is not predicting child outcomes. So I hope somewhat convinces you that's something about mood responding and dysregulation rather than just inattention, lack of literacy, or something else about reporting style on part of mom. Okay, so our next step is to take you out to the level of the home and talk a little bit about unpredictability in the context of the household or family context. And to do that, we're actually going to talk a little bit about family routines. And we're going to do that in the context of some work I've been doing in following these families during the COVID-19 pandemic. As we all know, a year ago, almost exactly, COVID really changed the world. And given that we're interested in the mental health of these children in these longitudinal studies, we felt it was really necessary, and actually so did NIH, that we implement extra COVID-specific assessments in these families. And so in May of 2020, we launched the first wave of COVID surveys in our study cohorts. I'm going to talk a little bit about findings from the preschool cohort, but I did want to note that we also have a paper under review looking at mental health in our adolescent and young adult cohort. And so I'm happy in the Q&A to talk a little bit about that if anybody's interested. And so again, this is critical for characterizing the COVID-specific experiences of each family. But for us, it also presented an opportunity to leverage this significant stressor to ask questions about unpredictability in the family context and child mental health. So this study, which came out just recently, had two goals. The first was to investigate mental health symptoms in preschool children during the COVID-19 pandemic. This was important because for those of you following the literature, which is really rapidly evolving, most of the work has really focused on adolescents, mostly on adults, some on adolescents, a little bit on older children, very little on kids, say, under the age of 5. And so that was an important aim in its own right. And the second aim was to test the hypothesis that maintenance of family routines serves as a resilience factor for children's mental health during the pandemic crisis. And just to orient you again, probably most of you are Californians or were here at that time. But again, this survey was administered in the month of May 2020, which is just after we were emerging from the first 7-week stay-at-home order that Newsom had ordered. So assessing the families just after they've all been home from that first almost 2-month stay-at-home order. I haven't talked a lot about the characteristics of these two cohorts in my lab, and I'll give you a little bit of information about this younger one. At the time of this assessment, the average age of the kids was 4 years. The predominant race ethnic identification of the parents in this sample is Latinx. 23% of the mothers and 40% of the fathers have a high school education or less in this group. 61% of these families' incomes are below the estimated living wage for their county of residence. Most of them are Orange County residents. The next two most common counties are LA and Riverside. 29% of these families are experiencing at least some food insecurity. 50% of these households had at least one parent working an essential worker, and 23% of the families had at least one parent who'd been laid off or furloughed as a result of the pandemic. I won't go into details, but in terms of mental health, we used a measure called the Preschool Feelings Checklist, which gets at child depressive symptoms. We measured child externalizing problems or conduct problems with the Strengths and Difficulties questionnaire. And I do want to note here that we also asked moms specifically to say for each of these conduct problems, things like tantrums and disobedience, whether that had increased or stayed the same increased, decreased, or stayed the same since the start of the pandemic. And then we did use a standardized measure of family routines, which gets at things like regular family time, meals together, and things like bedtime routines. Oh, and moms reported the extent to which these had been in place since the start of the pandemic in their household. What we found was unfortunate, but consistent with what we know is going on with the mental health of older individuals in the US. We saw really significant elevations in depressive symptoms in these kids, and also in conduct problems when we compared them to pre-COVID community and national samples. For the Strengths and Difficulties questionnaire, there are actually national norms for this. Here you can see some of those data visualized. So this is the answer to that question about whether these behaviors had increased, decreased, or stayed the same in terms of conduct problems. And what you can see shown in red on the right, for each of the 5 behavior categories, a sizable proportion of the mothers reported increases in these problem behaviors. Improvements were relatively rare, seen in gray on the left. And in some, close to 2/3 of the mothers reported increases in externalizing behaviors in at least one behavior category since the start of the pandemic, so very common. The good news is that in terms of family routines, it was the case that they appeared to be protective. So you can see plotted here that in blue on the left is the family routines inventory score for kids whose problem behaviors have not increased since the start of the pandemic for that particular behavior, versus those for whom the child had shown an increase in problem behavior, shown in red on the right. And you can see the higher scores among the children who did not show increases in problem behavior. So essentially, practicing more routines was associated with better mental health in the kids. And this is true also for depressive symptoms. I'm not showing the data here because they were analyzed continuously in both cases, but better mental health with more routines. And I'll wrap this piece up by just noting that the associations between routines and child mental health remained after considering child age and sex, family income, food security, dual parent household status, and also importantly, mom's depressive symptoms and perceived stress. These are, you could probably guess from the statistics I showed you about their level of socioeconomic disadvantage. This is a stressed group. Okay, so I'm going to wrap up by zooming back out and briefly talking about an additional index of unpredictability in early life. And while the mood entropy measures and the sensory entropy measures I've been talking about are, we believe, really potent predictors of child mental health, it's obviously not feasible for many to do such labor-intensive coding. And it's also the case that many don't have access to prospective data, but still care about early life influences on health. So with this in mind, we developed the QUIC, which is a 38-item questionnaire that assesses unpredictability in social, emotional, and physical domains in childhood. You can see that it has 5 subscales. The first is parental involvement and monitoring. This has items such as at least one parent made time each day to see how I was doing. Parental predictability includes items such as my parents were often late to pick me up, or for at least one of my parents, when they were upset, I did not know how they would act. Parental environment, there was a long period of time when I didn't see one of my parents. Physical environment, I moved frequently, and then safety and security, which has items such as there was a period of time when I did not feel safe in my home. So that's kind of an overview of this questionnaire that we developed. And in this initial validation paper, we show that it has really strong psychometric properties and also strong predictive validity. So in this paper, we show that the quick predicts depression, anhedonia, and anxiety in 3 independent cohorts, one of adult women, one of male and female adolescents, and then the third was a group of adult male veterans. We also documented, importantly, that it predicts mental health beyond a range of other indices of early life adversity. So we examined in the adolescents, which are the adolescents in my lab, we're able to look at prospectively characterized adversity in childhood, and the quick still predicts. But in the other 2 cohorts, using similar self-report retrospective measures, we show that it predicts beyond childhood trauma and also childhood socioeconomic status. And the childhood trauma one is significant in the male adult veteran cohort because that cohort had much higher rates of trauma exposure than our other 2 cohorts. So I'll end by just talking a little bit about where we're going with this in the collaboration that we have with the group at UCSD and the San Diego VA who are looking at this in veterans. There are new findings there that show that the quick is a really strong predictor of post-deployment mental health status in veterans. So they have data pre and post-deployment and 10 years post-deployment. And this is predicting, again, beyond childhood trauma exposures and other things, how these vets are doing. They've also linked the quick now to mental health and psychopathology in a group of treatment-seeking veterans who are not quite as healthy as the post-deployment group. We've now translated this into Spanish. I just, in my lab, wrapped up a Spanish validation study where we looked at this in 300 Spanish-speaking individuals. And what's exciting is that the associations with mental health outcomes look really similar to what we're seeing with the English version, which is exciting. A group at the University of Amsterdam recently also completed a translation study into Dutch and have nice findings, and our Finnish colleagues are just embarking on their translation study. I'm not going to go into it, but if anyone has questions about use in younger ages, we are working in the lab and validating a parent report version that can be used down to infancy. And with Rena Eid, who's at Penn State, she's a really interesting cohort of children who whose mothers have a history of substance abuse. And she's working on validating down for self-report use to 8 years old. Right now, you can only use it it's validated down to self-report at age 11. And then the last thing that I think is perhaps most exciting to me, because I've showed you a lot of quite basic work, is thinking about translation. And most recently working again with Hal Stern at UCI, we developed the Quick 5, or as someone is voting to call it the Quick, which is a 5-item version of this scale. And I can report it has also great psychometric properties and predictive ability, which is great. And so this is really suitable for settings in which time is limited, including clinical settings, because one goal of this group is really to try to translate this. And we've been in conversations with Children's Hospital of Orange County. CHOC is already screening for ACEs, or adverse childhood experiences, with the PEARLS, which is a more conventional measure of early childhood adversity, the one that's advocated by our current California Surgeon General, Nadine Burke-Harris. In any case, they're already doing this screening at every well child visit. And we've been discussing with them the possibility of adding the Quick 5 to their screening registry, which would be really exciting, because this is going to allow us to examine early life unpredictability and health in hundreds of thousands of Orange County children. So that would be a nice complement to the ongoing work we have with our cohorts. All right, so in the interest of time, I'm going to end there. I want to conclude by thanking my team at Chapman. This is a lot of work to run all of these visits and wrangle all of these kids and families. They've always done excellent work, but have really risen even more to the challenge amidst what's going on with the pandemic in the past year. I want to acknowledge my funding sources and also the intellectual collaborators on these projects. This really is a team effort, and it really benefits from everyone's unique expertise and perspectives. And last, I want to thank Chapman, who's really supported my program and provided a home for the work with these longitudinal cohorts. So I'll end there.
Thank you so much, Dr. Glynn. I don't see any questions in the chat, so I'm going to invite audience members to go ahead and unmute yourself and ask a question. I see Dr. Jenkins has her hand up, so why don't you start us off, please?
Great job, Dr. Glynn. Thank you for an excellent talk and for taking your time to share your work with all of us. This is really interesting and important work. I have a bunch of questions, so I'm going to try to merge two into one so that I can be efficient. Can you talk a little bit about what interventions in this area might look like? I can guess from, looking at some of the items in the quick what, we might expect for interventions to look like. But what about in the context of, mothers playing with their children? What would a, kind of like a good play session look like? And then also for those rodents who are in the LBN category, they were more unpredictable in their behavior, but presumably there's some benefit to why they're doing that, right? They're in a harsher environment, and so they're trying to adapt in some way. And so I could imagine that in some situations, interventions to just tell them to, if we could intervene, just be still and nurse for longer wouldn't be as helpful. So my question again, just to summarize, what would interventions look like kind of in the context of mothers playing with their children, but also in the context of when environments are unpredictable and certain kind of like behaviors might be adaptable, how interventions might play a role there.
Right, great questions, and I'm glad you repeated it because I wrote it down the second time, so I make sure I do both. So to your first question, what would interventions look like? I think, like you said, some things are easy, enhancing family routines, but when you're talking about the micro level, I suspect you're asking about mostly the sensory entropy or sensory signals.
Yes.
I can tell you a couple of things about that. One is that it's hard to see. So if I were to show you videos of the humans and have you tell me which is the one that's a high entropy mom and which is a low entropy mom, unless mom is catatonic, and someone hasn't asked this question yet, but could there be entropy that's so low that it's not good for development? And we can talk about that. But in general, within this normative population, it's not really easy to see. And what I can say, so giving specific directives is going to be difficult. But I do know that there is a pretty strong literature on enhancing parent-child behaviors in contexts like that I think could be applied here. And it's something we'll definitely look at. The other intervention, which has to do with structural manipulations, is actually socioeconomic status. And I think if you think of the fact that that really is a model, some way of poverty, we're working with, so Greg Duncan and Kim Noble, I don't know if anybody, Greg Duncan's at UCI, Kim Noble, Harvard maybe, have this intervention study, national intervention study, where they're giving money to mothers who live in poverty for the first, I want to say 2 years of their kid's life. Every month, they're either getting $30 or $300. And they have mother-child interaction videos. And one of the things we're going to do is work with them to code this, because my suspicion is that it may be doing things like reducing cognitive load and stress on mom that is going to facilitate interactions and organized behavior. So that's a different type of intervention that's more at the societal level, but I think that's something to consider there. In terms of your question about adaptation, Right. So it's always fraught when you say something is a developmental trajectory in a certain context is necessarily negative, right? It may be the right thing for that organism in whatever context they're in. And I won't necessarily say adaptation because my former evolutionary biology postdoc would kill me for using adaptation incorrectly. But, I think to the Steve Granger findings with what may be accelerated maturation of the unsnip, we need to look at trajectories to really say, is that accelerated maturation? But that's what the sort of conceptual background to that is the idea that accelerated maturation of that circuit may in fact serve the child or organism who that's developing in this adverse, unpredictable environment, right? There are trade-offs there, probably, but I mean, I think you're right. What mom is doing and what the consequences are may in fact be enhancing probability of survival or something else in that context. Yeah.
Thank you.
Others, if you have questions, please unmute yourself and feel free to ask.
I had a question. I don't know, this is probably a really basic question, but I was wondering for like the quick 5, why is it more beneficial to do that in like a doctor's office rather than like in a school setting? Because if a child is able to build a connection with, let's say, a teacher or something like that as an adult every day in their lives, why could it not be used in that situation for like more frequent checkups? Because I know, at least know a lot of my friends who didn't grow up with a lot of money never really went to the doctor once a year.
That is a terrific question. And what I can tell you is in this collaboration, we are also part of that screening registry I mentioned is not just at Children's Hospital of Orange County, but it also connects to the school districts in Orange County who are also screening for ACEs. So that's a terrific question. And you actually just channeled, we have a grant under review with the California Precision Medicine Initiative, which again is our Nadine Burke Harris's baby about early life adversity. A component of it has to do with screening, and you just got what some of the reviewers on the first round said to us, which is you're going to miss kids by just looking at well child visits. What about other contexts? So I think you're right, and I think that school is a great one because it will have the ability to access more kids, and I hope that we get it funded. We should be knowing any moment and push that agenda forward.
Well, I hope you do get the funding.
Thanks.
I had sort of a comment really. First of all, it's absolutely fascinating, and it's amazing to me that you've been able to find such a robust predictor. So, and the idea that it's not even a higher moment, it's entropy, which I don't know how to characterize, but it's not like a simple mean or some other, simple boiled down predictor. I know that in some EEG brain research over the last few years, they've been using something related to entropy, which is compressibility as a predictor of brain state. And it struck me when you were describing that predictor might actually work very well on your data. So you basically, it's very similar to entropy, but it reveals some things that entropy doesn't. You try to compress, you take your data and you basically try to compress it like making a zip file. And the measure is based on like the smallest lexicon that you can find that adequately describes the data, which is what zip does, which is what Lempel-Ziv encoding does. So anyway, it just struck me that might be really an interesting analysis to try on your data.
Great, yeah, that would be really exciting. And I was actually gonna reach out to you because I've been thinking more recently about EEG and P300 in response to novelty. And I haven't thought about EEG in a long time, but I wonder whether that threshold could possibly be set by some of these inputs in early life related to predictability and detection of novelty. So
yeah, and I think it would be really neat if this measure that has been used on EEG data could be used on what really are behavioral data.
Yeah.
And be, you know, applied just as well to those kind of data. I don't know. It just struck me that it might be really interesting.
That would be great. Let's talk about it.
If I may follow up quickly on Aaron's thing, one of the grad students and I have a paper now under review where we use this Lempel-Ziv. I mean, we developed a measure of compressibility based on this Lempel-Ziv thing. So just, yeah, happy to share that code and
Great.
if you need it.
Great, thanks.
Any final questions for Dr. Glynn? Okay, well, thank you first to Dr. Glynn for an incredible presentation, and thank you to everyone for coming today. Have a great rest of your week.
Thanks so much.
Thank you so much.
Thank you.